A very warm welcome to our webinar today. I see we have attendees from all over the world. That's great. My name is Jeanette or Jeany Prinz. I'm heading the life sciences team here at KNIME, and with me is my colleague Temesgen, who's also part of the life sciences team. Hi, Temesgen.
Hi, everyone.
Temesgen and I both have a background in bioinformatics. We're also both based in Berlin. So that's why we are today doing the webinar about gene expression analysis with KNIME Analytics platform. Before we start, just a little bit of housekeeping. So the session will be recorded and it will also be available on YouTube afterwards. We will send out a follow-up email with all the links, also the slides that I'm showing today. In the end of the slides here, I also have some promotion codes, so you don't need to scribble everything down. That will also be part of the email. And then last but not least, use the Q&A section to post your questions. This is actually how it looks like, right? We have that little thing here in the middle is the Q&A. And you can also upvote your favorite questions. So if you hit the thumbs up button, then it means that you think that's a very interesting question. And then we really make sure that these questions will get answered. One very brief slide about KNIME. I think those of you who joined a few webinars or trainings probably know that slide. So the KNIME software ecosystem basically consists of two parts. One is the KNIME Analytics platform with all its integrations and also extensions either provided by KNIME or through the community or our partners. And the KNIME Analytics platform is basically there to create data science. And that obviously starts with gathering and wrangling your data, and then you bring it into a shape where you can then visualize your data or create a model. Once you really want to deploy that model, that means you want to really bring it into production. That's where our commercial product, the KNIME server, comes in. That also allows you then to consume and optimize your model. So what you can, for example, do is you can create a web portal application. And this is also what we're going to see today. So the focus of the talk will mainly be in the open source KNIME Analytics platform, but I will also show very briefly how you can actually create such a web application in the KNIME web portal. So I will start with a little bit of an introduction to gene expression analysis with KNIME, a few slides. Then I will go into the demo of the workflow in the KNIME web portal, and then I will show the same workflow that's underneath the hood in the web portal in the KNIME Analytics platform. And then we will end with a Q&A where Temesgen and I are happy to answer your questions. So very briefly, I don't go too much into detail. Gene expression is basically you have the information from a gene is used in the synthesis of a functional gene product, mostly a protein. And the first step in gene expression is always the transcription, where the DNA is transcribed to RNA. And then you can actually use that RNA to quantify how much of a gene product is synthesized. And in order to do that, you can reversely transcribe that RNA to cDNA, and then you can sequence that cDNA in a genome-wide manner. I think we all, or many of those who are actually interested in that topic, know that sequencing become cheaper and cheaper in recent years. So that's really a way of quantifying of how much gene product you have in a genome-wide fashion. What we're going to do today, right, what's overall, if you want to do a differential gene expression analysis, your goal is often that you want to find the gene products with an increased or decreased synthesis under two different conditions, right? This could be, for example, in individuals with a particular disease. What we're going to look in today is we're going to use RNA-seq data from tumors and matched normal tissue from 3 patients with oral cell carcinomas. And that is based on a paper here from +1. Yeah, so you also here have the DOI if you want to follow that. Overall, if you want to do a differential gene expression analysis, what you basically do, right, you map those read counts that you get from sequencing back to the genes, and then you want to find the RNA-seq counts of genes that change significantly between those two measurements. And in our case, again, those measurements would be the tumor tissue versus the matched healthy tissue. The problem with that is often that if we do such an analysis, we find a lot of genes that are differentially expressed. And now what we actually want to do is we want to find the genes of interest. So we want to narrow that set down. And we can do that by investigating the functional annotations of those genes, or we can also look into genes with similar expression patterns, because also similar expression patterns often hint to a common function. And that's exactly what we're going to do today. So this is the workflow we're going to use. That's actually based on a blog post that you find in the link below. Again, we will share the slides in the end, so you will have the link and everything that comes with it at hand. I think one important thing here is what I basically want to show in that webinar is not necessarily a best practice about how to do gene expression analysis. It's more about to harness the power of KNIME, which I think the most important thing, or one of the coolest things in KNIME, is actually its openness for other tools. So what you always can do with KNIME very easily is mix and match technology. And that's exactly what we're going to do today. So we're going to use, for example, KNIME's R integration. We're going to use Google's BigQuery in that workflow. And we also use customized shared components to perform a gene expression analysis. That's exactly what I just mentioned, right? One thing about KNIME is that you can just easily mix and match technology, and that can range. You can just gather data from different external sources, which could be, for example, from Google Drive or data from a REST endpoint. Then if you want to visualize the data, KNIME has a lot of in-house JavaScript nodes, which you can use to do easy visualization. And if that's not enough for you, you can then, for example, also reach out to Spotfire or Tableau. The same holds for our machine learning nodes. KNIME has a lot of functionality already built in for that. And again, if you want to have more, you can actually reach out, for example, to H2O. And then if you want to have a fast execution of those models, you can run that in distributed or cloud execution. And again, if that's all not enough and you want to, for example, have more customized visualizations or also a very particular machine learning library that's, for example, available in Python or R, you can also reach out to those two programming scripting languages. And this is also what we're going to show today. So we're going to use R and use our favorite R library in KNIME. And that basically allows us to combine a lot of different technology, but in a way that we can produce one reproducible workflow in KNIME that then we can obviously share, and we know what kind of sectants we used. We can annotate that workflow very easily, and that harness a lot of power. The workflow that I'm talking about today is on the KNIME Hub. So if you go under hub.knime.com, you will find a place to share knowledge about workflows and KNIME nodes. The workflow that I'm talking about today is also here available under this short link. The nice thing is you can just go follow that link and then open the workflow in your KNIME analytics platform automatically. I will talk a little bit more about the hub in a few minutes. Just for those of you who are very new to KNIME, I assume a little bit that you already have heard about some concept, but just that we are all on the same page. KNIME is a tool to create data science workflows, right? And the smallest part of that, the workflow, is a node. So in KNIME, we have nodes that perform tasks on data, and that could, for example, be a simple tasks such as reading a file or grouping data or partitioning data. And then we combine those nodes into more complex workflows just by putting them together. And those workflows then solve a particular task, right? In our case, our task is to find interesting genes from a gene expression analysis. In between the nodes and the workflows you see here is the word component, and the component is really a little bit a thing in between. A component is basically an encapsulated little workflow snippet, and that allows you to keep your workflow a little bit in order, right? Because you can just hide and encapsulate functionality. So that's one of the things that's actually very useful about a component. But a component is actually way more powerful, and we will see that in a second. And at the same time, you also see that a component looks a little bit like a node, right? From the size. And often a component performs a particular task as well, right? You want to encapsulate a functionality that is designed to solve a particular problem. And one cool thing about a component is, again, as I said, it encapsulates a set of nodes into a component. And one thing what you can actually do with components, you can also parameterize components. So if I add into a component, for example, here a column selection configuration, that shows up, if I then do a right click on my component, that shows up as a configuration in my component. And that's what I can do with that is basically create little KNIME nodes that have more workflow snippets under the hood. But on the whole run, they basically behave like a normal KNIME node, and they also look like it, right? So that's why the components, if we look back, are in between the nodes and the workflows. Because on the one hand, they are like a workflow because they have a little bit of a workflow snippet underneath the hood. But on the other hand, they also look like a node, and we can also make them behave like a node. And what you, for example, also see in that workflow here is one of our JavaScript views. And what I can always do with such a JavaScript view is I can do a right click and then look at the associated interactive view, which we will also do today. And I can actually also do that with components. The cool thing about the components also is if I have multiple JavaScript-based nodes inside of my component, then I also see those in an associated views. So I can, with components, I can combine views and I can create composite views. And the cool thing about that is also that the selections that I do in one graph, they get transmitted to the others. So for example, and we will also see that today, if I select something in that scatter or volcano plot in the view here, that gets automatically selected in the table as well. And I don't have to do much for that. So as long as they are working on the same KNIME table, they can immediately talk to each other without me having to program anything. So I think that's already very, very powerful. And then basically one step further we push that is that you can actually share components. So it's not only that I create a component and then I need to do that over and over again. What I can do now is I can save component instant into my KNIME workspace. And I can, for example, just share that with myself. And here the use case would, for example, be if I'm connecting to a database in a lot of different workflows, and then I extract a particular set, a particular table, and then I do a row filter or whatever. So I have a very particular pattern that repeats over and over again. I can just share that with myself and reuse that in different workflows. So I don't have to do that pattern all over again. So that saves me obviously a lot of time. Plus, now when I want to change something, I can actually change that in one component. And as those other components are linked to each other, I can just update that. So that also saves me a lot of time. So if I actually want to save a component or share a component, what I can do is I just do a right click on that component, and then in the configuration, I go share, and then I can either save it in my KNIME workspace or on the KNIME server, or on the KNIME hub for later use. And we will actually also see how we can then use those components from the hub today. One thing also to mention in that context is that shared components are read-only instances of a component. So you can't just make changes. If you want to make changes, I will actually show that to you today as well. And then we from KNIME realized the power in that a little bit, because we thought, hey, that's super cool. Everyone can actually create their own little nodes and share that. So we also started to create those shared components and share them with the community. So those you can find A on the example server and B on the KNIME hub. So this is what you see here. So if you go in your KNIME Analytics platform in the upper left corner, you see that example server. And then under 00 components, you find a diverse set of topics ranging from automation through guided analytics, through text processing and time series. And obviously most interesting for the audience today are the components regarding the life sciences. And again, we have different components there. They're mostly concerned with gathering of data. So you can, for example, gather data from Campbell or from the European Nucleotide Archive or from PDB. And the component that we will be actually using today is a pathway enrichment analysis component. That was also provided here from Franzi from the life sciences team. And that allows us to find biological pathways that are enriched in a set of genes more than would be expected by chance. This is based on the Reactome web service that we will, so we actually see how to use that today as well. In general, if you want to learn a little bit more about components and how they are sharing or how you can share and reuse them, there's also a blog post by Michael that I would definitely recommend to you to have a quick look. Another set of components that are interesting, I think especially also in the life sciences field, are in the visualizations category. So for example, for those of you who are in the bioinformatics field, I think the sequence logo would be of interest. And then what we actually gonna use as well today is a component that allows us to do a hierarchical clustering and then show a heat map of the clustered data alongside the dendrogram of or resulting dendrogram from the hierarchical clustering. So that's basically one aspect. How we can mix and match technology is via shared components, and that we will see that in the workflow today. Another thing that we're going to see is our R integration. So with KNIME, you can run any R code. It works with existing R installations. You basically just point to the path of your installation. We have nodes for many tasks. For example, if you want to have a particular view or visualization, you would use the R view. And in order to make that work overall, all those nodes, you first have to install the package rserv. If you are a Windows or Linux user, that's enough. If you are on Mac, you have to additionally install the package Cairo, and then you are ready to go. I think one of the most valuable nodes in that set of nodes is the R snippet nodes. It's very generic and basically allows you to execute any kind of R code. And this is also the node that we will use today. Very handy is that there's syntax highlighting, which makes your code a little bit more easy to read. You also see the columns that come in from your KNIME table, and then you can just easily double click in it and just use it. Then here you see your R workspace with the variables that are created, and then you can evaluate the whole script or just a selection, and then you see the result in the R console output. And obviously that allows us to use different R libraries, right? So there's a lot of stuff already provided in R, which is cool. And through KNIME, we can actually harness that and just create a reusable KNIME workflow that is based on those. packages as well. So what I always did also in the past before I was working for KNIME, like a typical KNIME workflow looked for me that I did a lot of the data munging and the data wrangling and putting the data into shape in KNIME. But then I used a particular package where it was very easy for me to use in R. And then I did the visualization, for example, again in KNIME. So this mixing and matching, again, I think is very powerful, and you can just create a easily reusable workflow for that. Today, we're going to use the R package Edger. It's a bioconductor package for differential gene expression analysis, and it's particularly useful for RNA-seq data. And we will use it to adjust for different things, sequencing depth and RNA composition. So it's a little bit a normalization step basically, right? Because it could, for example, be that highly expressed genes can cause the remaining genes to be undersampled. So that's always something to take care of when you do in differential gene expression. We will not take account for differences in gene lengths. That's something you'd sometimes want to normalize for, but as we have data where we have the same genes we're comparing, right? So we have the tumor and the matched tissues. We don't have to account for that. And then we will use the normalized data to fit a GLM or generalized linear model. And then we apply a likelihood ratio test. What that brings us in the end is a log-fold change value for each gene. So that basically allows us to estimate the magnitude of change between the two conditions, right? And again, the two conditions would be healthy versus tumor tissues or diseased patients. And so the log-fold change allows us to find the magnitude of change. And then we also have an associated p-value with that, which allows us to assess the significance of the results. We do that for a lot of genes, right? So in our case, you will see that we have over 10,000 genes. So we obviously have to correct for multiple testing. In our case, we do that with a Bonferroni correction, which is obviously very stringent. If you have other favorite choices like Benjamini Hochberg or whatever, you can obviously do that too. But that's really then up to you and your choices of data analysis. One small side note for the R packages, again, we use today Edger. You can use any other package that you like. I think one really nice blog post in that context that also uses our R integration and a different R package called Data2 is from Temesgen, who's also here with us today, and he did a blog post about using KNIME for microbiome analysis, and you can also find that workflow on the hub, as well as there's a nice blog post that I would definitely recommend to check out. Again, all those links will be made available for you after the talk today. Okay, so maybe just a very quick recap. What we did so far, what we learned so far was we learned that we want to do a differential gene expression analysis, and for that we want to mix and match technology. And what we did so far, what we learned so far is we can use R in our favorite R package to find differentially expressed genes. And then we can also use shared components to analyze that further. And then in one last step, what we're going to do, or what we want to do, is to use Google BigQuery. And here our goal is to find compounds that target the selected gene products. And in our case, we want to query bioactivity data from the database Campbell. And for that, we use the KNIME Google BigQuery connector in combination with the KNIME database nodes. Obviously, there's also another way of doing that. And I think that's maybe also something that the KNIME users among you experience already. There are often many ways of doing things in KNIME. Here, I actually wanted also to showcase how you can use Google BigQuery. Another way of doing that would also be a shared component. We saw that before. We do have a Campbell component where we can actually also query bioactivity data. We have another blog post about querying the Campbell REST services. So you could also just do that yourself, for example. So again, there are always many ways of doing things in KNIME. We will use Google BigQuery. If you want to learn a little bit more about Google BigQuery, we have a very nice blog post by Emilio that is basically just a tutorial of how you can get your data into BigQuery or extract data from BigQuery. If you are more interested a little bit more in the scientific side of things, there's a nice blog post by Martina and Greg, which is about the interactive exploration of scientific data sets, more specifically the SciWorker open data in KNIME. So check that out as well. We always have a lot of material, but I think there's also a lot of cool things you could do, right? All right, so with that, Temesgen, are there any urgent questions that should be answered? If not, I would move into the demo on the web portal.
No, you can proceed.
Perfect. All right, so just a quick reminder, what we're going to do now, we're going to quickly see the workflow as a web application, and then we will actually look into the underlying workflow in the KNIME Analytics platform. And again, in order to do the analysis, you don't need to have access to the server. I think there's just one little addition, which is just really nice because you can easily create those web applications. So this is how we start, just a very quick introduction of our workflow. And now in the first step, what we want to do is we want to actually select the data under the two conditions of interest. So I'm going to select my file, one of the patients with the disease and one without. And then I'm going to hit the next button. And what's basically now happening under the hood is a KNIME workflow is running. Again, we will have a look at that particular KNIME workflow in a second. And that KNIME workflow is using the R package edger, and then we create an interactive visualization using the KNIME node. So that is basically a component, but we're going to see that in a second. Again, differential gene expression analysis, the output is the fold change, right? So it's the magnitude of change between those two conditions. And this is also what you see in this typical Volcano plot here, where we always have on the x-axis the fold change, and then on the y-axis I have the false discovery rate. False discovery rate is just the Bonferroni corrected p-value, multiple testing corrected p-value. We have that here in a logged scale so that we can actually see the changes equidistant from the center. I already said that before. That view is interactive, or maybe one thing before I talk about interactivity, I colored also here the data points. Each data point is a gene, and it becomes the blue color if the false discovery rate is bigger than 0.01, and orange or whatever that is if it's smaller than 0.01. What I have also on the bottom here in my view is a range slider where I can filter the fold change. So if I, for example, now do this, I actually see that in my plot, my data is gone as well. So again, that's exactly what I said before. The plots are able to talk to each other. I can also select data, for example, here in my plot. And if I allow here show selected only in my table here, then I see, aha, those are the genes that I selected before. And if I just want to have a little bit additional information, for example, I see the fold change again, I see the original p-value, and I see the false discovery rate. If I now click on next, what we're going to do now is a hierarchical clustering. And so as I already said, We want to see a heat map and alongside a dendrogram. So what happened now under the hood was a hierarchical clustering of the read count, the normalized read counts. And the idea here is a little bit what we want to see is if genes have very similar expression levels, they often also have a very similar function, right? And we want to actually, in the end, we want to find genes that may have something to do with each other, maybe work together in a particular biological pathway. And if that particular pathway is disturbed, that might be a hint that that is the reason that's actually giving rise to the phenotype of the disease that we are looking at. And that's a little bit what I see here, right? So we have, for example, genes here that have an overall low baseline of expression, and then they have an increased expressions in the patients with the oral cell carcinoma. But we also see, and that is indicated by the color, so the blue means a rather low expression, and then red means a higher expression. And we also see here, for example, a set of genes that have an overall higher expression in general. Again, the whole thing is interactive, so I just selected something here and that got automatically selected in the other plot as well. And the nice thing, what I also have below here is a table where I can then look a little bit more into details about my genes that I'm looking at. So now instead of only the name, I also have the ensemble gene ID, which is always better to work with anyway than just the gene name. And then we also have a description of the gene under a consideration. So here, for example, we have the gene, the calmodulin, which binds calcium, or here we got collagen, which is a structural compound of an extracellular matrix. Even if we scroll down a little bit, then we have here another gene where it's already said that it may play an important role in the progression of epithelial malignancy. So that already could maybe ring a bell that we are on the right track. What we're going to do now in the next step is we're going to perform pathway enrichment analysis, and then we're going to see the result here, and we see also on top like the first two most significantly enriched pathways are on the one hand collagen degradation and degradation of the extracellular matrix, and it is actually known that interactions with the extracellular matrix are involved in tumor invasion and metastasis in oral squamous carcinomas. So we can select that now, again, see a little bit additional information, and then we can click next. And what will now happen to it, I think I will actually show that in my KNIME Analytics platform. We're already a little bit shorter on time, is we will query Google BigQuery and find targets that target the gene product of interest. But first things first, maybe let's take it slow here. So that is actually the workflow that we just saw the results from. So this is KNIME Analytics platform, and what I said before, right, this little thing is a component. So if I do a right click here and I click on interactive view, that now opens exactly the same interactive view that I just saw on the web portal. And so by creating, right, so that should all ring a bell for you. And so by creating such a composite view through a component, at the same time, I can just easily create a web page on the web portal. And the only thing that I have to do after I created that workflow with those components is to do a right click here and then deploy to server, and then I have to save it first. Right click on the workflow that I'm working at right now and then deploy to server, and then I can just deploy it on my KNIME server. And what I can then do on the KNIME server is a right click and then open in web portal, and this will then directly point us to the web page where we started today. Okay, but So we're going to reverse everything a little bit. So now we actually want to look into the workflow and how we created those web pages or composite views. So what we start with is the interactive view, if you remember, where we uploaded the data. Another way of putting that data in here is through a configuration. So if I do a right click, maybe zoom in a little, I don't know. What you see here is just a configuration, and I can just browse and add my data again. Here what I did is I used a little bit of a trick. So for those of you who are new to KNIME, don't worry about it. For those of you who are already a little bit advanced with KNIME and maybe used quick forms in the past, pay attention. So if I go do a double click inside of a component, then I can actually, so control double click allows me to look into that component and see the encapsulated workflow snippet that's in there. And here I used a little bit of a trick. So what you see here is the configuration node. And if I just give the configuration, I do a right click, then that's actually the thing that's going to be used. However, if I use that workflow on the KNIME web portal, then that value gets overwritten by the variable that I give here through the file upload widget. So this way I can let those two talk, and I can easily override it. And now I have the functionality that either if I do a right click on the component, just to see that again, right click, then I can do configure and just give the data, or what we saw before, I could also use the view on the KNIME web portal. But that's a little bit an advanced trick. Don't worry about it if you're relatively new to KNIME. What that now allows me is I have my data in KNIME, and now I can actually use that count data in my R snippet. Maybe just have a very quick look at the data. That's actually also one thing that I always like about KNIME. You're always very close to your data. So if you do a right click, then you can actually look at the table, and then we just see the gene name, and we have the patient ABC, and then we have the control. And now we use that in our R snippet. If I do a right click again, I see my R snippet, what you saw before as well. Most importantly here, I can just load in my favorite R package, in this case, the R package edger. And here I have also the columns that I just saw in the KNIME table. I can interact with those columns pretty easily. So I can just do a double click, and then I can actually already use that as a data frame and work with that. And again, what we're basically doing here is we're fitting a generalized linear model. We're normalizing the data a little bit, and then in the end, what we get is our fold change and the p-value already Bonferroni corrected. That's what you see here. So that's happening in that R snippet. And what we're going to do now in the next step is we want to create that visualization that we already saw before. So we want to do the typical Volcano plot, which is in the end just a scatter plot. And then in addition, what we want to do is we want to have that little filter here with that fold change. And we want to have a table where I can select something in the table, and that gets automatically selected in the scatter plot and vice versa. So again, if I do a control double click, I get inside of my component, and here I already see the blue color of the node indicate that that's a JavaScript view. I already see that I have here that interactive range slider, and here I obviously just say I want to have the log fold change as a label, and then this is actually the column that I'm using the range from. And then I use my scatter plot to display the log fold change versus the logarithm of the files discovery rate. And then I use the table view to actually display a little bit of the additional information, including the row colors and the row keys. And in the row key is actually the gene name. Here is actually the coloring happening, right? If you remember, I gave a particular color to the ones that are significant. And for that, I actually did a binning before on the false discovery rate in order to have discrete values here. And that's already it. Again, in order to make those two talk to each other in that interactive view, I don't have to do anything. By default, that interactivity is already enabled. I can disable it if I click here on activity and then enable selection, but that by default that's on. Here, this filter is in addition there because I want to have that the interactive range slider actually talks to my filtering that I select. But apart from that, all the interactivity is already done. So that's it to create that component. What we're going to do now in the next step is creating that clustering and heat map visualization. So again, all the visualizations that we saw before, they are also in that workflow because that is actually the workflow that is running under the hood when we saw the web portal. And this is the visualization that we see here. We have the heat map, we have the dendrogram, and it's actually sorted by the clustering. And we have that table with that additional information, which is very valuable, right? Because here you actually see already what the genes are doing. And as we already narrowed down the set to a few more interesting ones, we can now really start looking into the annotations. So how do we do that now? That's actually where the shared components come in. So if I go under my KNIME Explorer and here under examples, if I click on show examples, that opens the example server. So also for those of you who are relatively new to KNIME, the example server is a quite useful tool to get started. So we have a lot of examples for very different use cases. Today, I actually want to talk about the components. So if you go under 00_components, you see exactly that image that I showed before in my slides. So we have all those different components from automation and through life sciences and also the visualizations. And you already see here, I have that component in there. I will just delete it right away. And now what I can do in order to reproduce what I just did is I can just do a double click. Oh, no, that's not what I wanted to do. I wanted to drag and drop it in my workflow. So I can just drag and drop that into my workspace. And now I get a read-only instance of that shared component. And now what I can do is I can click or do a right-click configure. And now I can actually tell my component what is the data that I want to cluster on, and I will use the normalized read counts for that. So I do not want to cluster on the names of the genes or anything like that. So I only want to use the read counts, and now I do execute an open view. And now I see that view from before. So I have again that dendrogram, which is interactive, and I have the heat map on the side. And what you already see here, right? I don't have that additional table, but I used a trick basically. So I have here not only that dendrogram and the heat map, but I also have that table view. And the trick is, right, you can nest components. So you can actually create components within components. So you can be very meta about it. And this is what I did here. So I just added that additional table in a component. And that allowed me then to create that interactive view where I have that table in the end as well. So this is how I can use shared components through the example server. Another way of using shared components is through the KNIME Hub. So if I go under hub.knime.com, I can, for example, type in pathway enrichment. Already mentioned that before. And then I actually already see here pathway enrichment analysis, and I see, aha, that is a component. I can now click on it, read a little bit more about it, make sure that that's actually what I want to do. I know that that is actually what I want to do. And now what I can do is I can drag and drop that into my workspace. And now I have that additional pathway enrichment component available. I just delete the one that I used before, and I can combine now those. Little bit pro tip: if you just select the nodes that you want to connect and then Control-L, those get linked to each other. And now I have to configure it again, obviously. So what a pathway enrichment does, again, just a quick reminder, we had it on the slides before, is we want to see if we find a set of genes enriched in particular biological pathways. And for that, I obviously have to tell the node what is my set of genes. So I say here, aha, here's my ensemble gene ID. I click OK, and then I do execute and open views. And I see the view that we saw before. So I have here the collagen degradation, and then I see the genes that are associated with it. One thing that I mentioned before is that that is a read-only instance. Yeah? So you see here this little link here, that in the lower left corner of the component there's an arrow. And that means that that component is linked. So if I go inside of that component, I see everything is gray and I can't move things around. So that means I have a read-only instance of my shared component. If we just quickly copy it and paste it, just to play with it. What I can do now is a right-click component and then disconnect link. And then that component does not have that little arrow anymore. If I go inside now, I see, aha, I can move it around. So this is also a way of customizing your components, right? You can just use the components from the example server, either add a view, what we did before with the hierarchical clustering, or you can just disconnect the link and adjust the component according to your needs. So you can really customize your analysis by mixing and matching what we already have available there. And this way you can save yourself a lot of time. So what we did now, we already saw a few interesting pathways. Just a reminder, we had the extracellular matrix and the collagen, and I already mentioned that extracellular matrix interaction are known to be involved in tumor invasion and metastasis in oral squamous cell carcinoma. And now, as we basically figured out a few or a subset of genes that we think is interesting, what we want to do in the very last step, we want to look into compounds that maybe target those interesting gene products. And that's exactly what we did here. So in that last view, you see a tile view where we have all the compounds that target the selected genes of interest. How did we get there? Again, I do a control double click inside of my component, and I see here my Google BigQuery connector that allows me to extract the data from Google BigQuery. You obviously need to have a license for that. And if you want to learn more about how to set that up, I would really recommend those 2 blog posts. And then we just gather the data. Again, we query Campbell and the IC50 and KE values. And then in the end, we get, we extract also the SMILE string. And so this is the result of the SMILE strings. We generate an image from that. And now the nice thing is that you can use images in your tile view. So that's what I do here. So I say, okay, I want to have the compound ID, and I also want to have the image of my compound that I extracted. So that's pretty much it. So just a quick summary when we look at the overall workflow. Again, what we did is we started off with like 2 measurements that or two conditions of RNA-seq data. Then we use that in an R snippet to find differentially expressed genes. Then we created an interactive view in a component where we could interact with the data, and then we actually narrowed that set of genes further down to genes with shared function. And we actually also looked into the function through a pathway enrichment analysis. And then in the last step, we performed or we looked for compounds targeting the discovered gene products. Again, a lot of info, but my goal here was mainly to show you how you can use different pieces of technology in KNIME for differential gene expression analysis. You are obviously always the expert, so you know what your data needs are, but I think you have a lot of different tools at hand to actually perform what you really want to do. So I wanted to show you a little bit how many things you could do with KNIME.
So there were a lot of interesting questions. I tried to answer them live, not live, but using the Q&A. But there are a couple of questions I saved for you. Just one question, actually, I saved for you, Jeany, and the other one I will answer live. This is a specific question from Aaron Hart. He is asking, where does the gene annotations come from?
Yeah,
how are they generated? So I think you can answer that.
Yeah, that was an excellent question. So I skipped a little bit, a few things. So for example, what I'm doing here is also a little bit of a mix and match technologies trick. So what I'm using here is a GET request to query data from OpenTarget. So if you see here, that's actually the part where you see the interesting part. Is in the string manipulation. So if you click here, you see that I connect to the API from OpenTarget, and that then allows me to get the annotations, but also the matching to the different IDs. So again, we have a little bit of a mix and match technology that I skipped to keep it a little bit simpler. Thanks for the question, Aaron.
Okay, I will take over and start answering some of the questions. Oliver asked if when I change my R workflow, would I need to copy paste the code again, or could a node be pointed to the R file and automatically use the updated code? I actually used a similar technique before. So using the source command in R, you can actually point to your source code if you have R functions lying around, or if you have like a file, an R file, a source file lying around, you can point to that file using the source command in R. And then all the functions in that file will be available to use in your R snippet node or whatever R variant node you are using. Another question is from Joseph. He is asking, can you also do interactive 3D plots of gene expression data similar to the Volcano plots? We have a couple of visualization nodes that can do 3D plots. I'm not sure if they are as interactive as the one Jeany showed today, but I advise you to check them out. You can select, they should be interactive since they're Java script nodes, but I don't know how much flexible they are going to be.
And it's a 3D scatter plot in this case, right?
Yeah. So you can just look for 3D scatter plots or 3D plots just on the KNIME hub, and you'll be pointed to a list of nodes that can do that, and try them out.
Yeah. a new question actually coming in. Maybe I start with that one too. It's a good one.
Okay.
So if we have to use multiple technologies in KNIME, why should we use KNIME in the first place? We can use R itself for the entire functionality, right? So that is an excellent question. If you are able to do everything in R, why not do it, right? I think there are multiple reasons not to do that. A is reproducibility. I think those of you who use R, right, the odds are that you might then have a snippet lying around which you use with a particular parameter, but then next day you don't really know which parameter you use. With KNIME, it's very easy to annotate and to create reproducible workflows. Plus, many things are way easier, right? So I personally find it, especially that what I said before, right? The data wrangling, joining data, filtering data, all that stuff, or connecting to a database, way easier with KNIME than with R, for example. And again, so today the topic was a little bit, you can mix and match technology. With KNIME, you don't have to mix and match technology. We have a lot of built-in functionality. And the nice thing, obviously, is also you don't have to do that from scratch, right? You don't have to write your own code. You can just drag and drop stuff into your workspace, and that does save you a lot of time. But then again, right, if you're really happy with R, or you do have R snippets already that you want to reuse, might as well do that with KNIME as well.
Yeah. I was typing an answer, so why not actually answer it live? So Anton asked if I have a question, who should I email later? But actually, I strongly advise to use the KNIME forum so that actually your question might be also a question for others. And this KNIME forum is really active and vibrant. And the community there will give you an answer within, most of the time within a day. And then also the knowledge will be shared with others, right? So I advise the KNIME forum would be an excellent place to pose your questions.
And then also other people can learn from it, which is great. Yeah. And also just to make, it's a very excellent point, Temesgen, because I didn't mention that in my presentation. The forum is really a place to check out. And if there's someone posts a question and that does not get answered in a timely manner, we from KNIME really make sure that those questions get answered. So you will get your answer. All right, I would say we do one more question. We are already running a little bit late. Temesgen, do you have?
Okay. That one was not a question, just a comment, I think. There is one. Is moving data in and out of R nodes time consuming? So the answer is yes and no. So there are two techniques. You can use the R source nodes to create workspaces and use that workspace again. And there is a cost associated with that because you have to load data and save data. You have to persist it to disk. But you can also do that in just one node and then avoid multiple nodes, multiple reading and writing. On the other hand, it's not really. It wouldn't be worse than what you do with R. So there is the cost of saving your workspace and reading it back again, but you do this when you want to modularize stuff and have more readable workflow. Then you have to do the balance, you have to compromise between the readability of your workflow and the amount of time you spend reading and writing workspaces.
Absolutely. Maybe one more thing I saw that Temesgen, you already answered that, which is great, but it might be even worth showing. So someone was asking if one can rearrange the order of the composite view I actually meant to show that. So if you go, so I'm inside of the composite view now, and if you go here in the toolbar in the upper corner, you see that node usage layout, and here you can actually move things around and change the layout, and even put things side by side and things like that. So that's an easy way of changing the layout, because if I now look at it from outside, I see it looks probably pretty ugly because I did just a random layout, but I think you get the gist. But now I have those side by side in the table also there. That's an easy way to do that. And then maybe one last thing, which I think was an excellent comment. Oh, no, it's gone. It moved to answered. I think Eric's name, it was Eric saying that he often uses that different packages as a type of a sanity check, and KNIME then also makes the comparing of the results easier. So I think that was also a nice point. Thanks for sharing that. All right, good. Then thank you everyone for joining. It was fun, and thank you to Temesgen as well. And I hope to see you all in our next webinar. Bye-bye.
Bye-bye. Thank
Bye.
you.